And we've derived the final stage of cellular respiration. And it actually has two parts, which are the electron transport chain and something else called chemiosmosis. And this is where we're actually going to make the majority of our ATP. Up until now, you haven't really seen hardly any ATP being made. We made like 2 ATP during glycolysis, and then another one during the citric acid cycle, but hardly any ATP manufacturing. Whenever we've made ATP along the way, it's just been like an enzyme doing a reaction, and that's called substrate level phosphorylation. Substrate level because there's an enzyme, and it's just grabbing a phosphate group, grabbing an ATP, smushing them together, making ATP. So it's just a regular chemical reaction. But substrate level phosphorylation isn't going to give us the majority of our ATP. Instead, most of our ATP is going to come from these two steps, ETC and chemiosmosis, and they don't use enzymes in the same way. In order to have this whole thing make sense, it is important to understand where in the mitochondria we are doing this. And so if I was to draw like a rough sketch of how a mitochondria looks, it's got an outer membrane, and that's the oval, and then it has an inner membrane, and the inner membrane is folded upon itself to make all of these little fingers. And so when you have the two membranes folded, well, the inner membrane and the outer membrane folded like that, there's actually like several different spaces we could talk about. There's this space all the way to the inside that is inside of both membranes. There's another area that's in between the two membranes, or we could be talking about the space that's all the way outside of the mitochondria altogether. So this inner space all the way to the inside, that's the matrix. We've talked about that before. That's what the citric acid cycle is going to happen. There's the cytoplasm out here, and that's where glycolysis is going to occur. And then in between them is this gap, and this gap right here, that is called the inner membrane space. Okay, so we'll just say that the IMS, okay, the inner membrane space, and that's in between these two membranes. Well, the first thing we're going to talk about is the ETC, the electron transport chain, and the electron transport chain is inside of this membrane. So these two membranes have pretty boring names. It's just the inner membrane and the outer membrane. And so the ETC is inside of this inner membrane. And so everything we're going to draw is going to be happening inside of here. And there's not just one, there's like hundreds and millions of them, all stuffed within this membrane. So the way it looks is we've got, whenever I do this drawing, I usually draw my protein complexes first. And so it's not like just one enzyme. There are big old bundles of enzymes that are all together that are performing all of these different chemical reactions. And so these bundles of enzymes we call protein complexes because there's a whole bunch of proteins, they're all working together, they're all pretty close to one another. And so we say protein complexes. So we've got a total of 4 protein complexes. This is number 1, this is number 3, this is number 4. Protein complex number 2 is down here. So 1, 2, 3, 4. And then we've also got protein complexes that will bridge this gap, but they aren't really called protein complexes, they're not as big. And so this one up here is called the cytochrome C complex. And this one down here is called plastiquinone. I'll just put a Q for plastiquinone. And so the thing is that there is an actual physical gap in between these two protein complexes, but we need to carry things from one to the next, and we can't just like let it fly by magic or something. And so plastiquinone moves. It can move within the membrane to take it from here to there. Same with cytochrome C. It can bridge the gap to take something from here to there. And so as we look through this whole series of events, they're there to fill in the gaps so that we can actually get from one place to another. So it's going to happen inside of this right here. This is the electron transport chain, so the ETC. Inside of the electron transport chain is NADH is going to dock with protein complex 1. And when it docks with protein complex 1, it's going to turn back into NAD, and it's going to drop off the electron it's been carrying. So remember, this whole thing, the whole time has been about high energy electrons, and we're finally using them. And this high energy electron is going to travel from protein complex 1 over to plastiquinone, over to number 3, and then through the cytochrome complex over to number 4. And as it goes from one protein complex to the next, these protein complexes are going to use that energy within this electron to perform a service. And the thing that these are going to do is these are actually pumps. These are active pumps, and they pump protons. They're proton pumps, but they're going to do active transport. And so remember that you can't do active transport unless you have energy. Typically, we use ATP, but in this case, we're going to use the energy from this electron. And so when this electron arrives at protein complex 1, we're going to take some of that energy and use it to pump a proton to this compartment. This compartment is the intermembrane space, this one that we mentioned over here in between these two membranes. We're going to pump it up there. Well, if you think about it, you're like, well, why do we need energy to do that? The only reason we would need energy to pump a proton up here is if there's already a whole bunch of other protons. And if there's already a whole bunch of other protons, the protons don't actually want to go this direction. This is against the concentration gradient. That's why it takes energy. So we're pumping this electron upwards with the energy derived from that high energy electron. Now that electron is going to go through the plastic quinone. The plastic quinone is going to drop it off at protein complex 2. And at protein complex 2, the same thing's going to happen. We're going to use the energy to pump another proton across this membrane into the intermembrane space. Now the electron's going to go to cytochrome C. It's going to arrive at protein complex 4. And at protein complex 4, the energy, the last remaining energy in that electron, is going to be used to do the same thing. It's going to pump another proton upwards into the intermembrane space. And now that we've gotten to protein complex 4, the energy from that electron has been used to drive the transport of 1, 2, 3 protons. And it's no longer high energy. We've used up all that energy. It's now just kind of a low energy electron. And so we need to do something with it. But we can't just let it go. Even though it doesn't have as much energy as it used to, it is still something called a free radical, which means that it's an electron that's unbound. It will bounce around all over the place. It's like a bullet if it just isn't bound to anything. And it will cause damage. It could kill this entire cell. So we can't let the electron just go free. It needs a babysitter. And so the molecule we're going to use to babysit the electron is an oxygen. So the oxygen is going to bind with the electron. It's going to pick up some protons that are hanging around. And when you have hydrogen bound to oxygen, you're going to get water. And so we're going to generate water at protein complex 4. And this is the reason your body needs oxygen. Your body needs oxygen to simply babysit that electron so that we don't generate these free radicals within the cell, because again, they're very dangerous. So it's like kind of anticlimactic. You know, you need oxygen, you have to breathe. If you don't have any oxygen, your body starts freaking out. You know, if you've ever had a time when you've been kind of like in a scary situation where you couldn't breathe, it makes you panic. But all we really need oxygen for is just to babysit these electrons. It doesn't go through some sort of really complex process. It's just used to babysit that electron so you don't have any free radicals. And then it makes water. So for every molecule of NADH, we're going to be able to pump 1, 2, 3 protons. The other electron carrier, though, is FADH. We also had FADHs along the way. So same deal, FADH2 is going to come up here, except that FADH2, it arrives at protein complex 2. It can't bind to protein complex 1. It arrives at protein complex 2, and it's going to turn back into FAD. It's going to drop off its electron, just like which happened over here with NADH, and it's going to go from protein complex 2 up to plastic quinone and then join the party and do the same set of reactions. It's going to go to protein complex 3 and then to 4. But you might notice that it's a one-way street. You can't go backwards on this thing. And so NADH is going to get 1, 2, 3 protons pumped. FADH, it joins the party at protein complex 2, which means it missed number 1, and you only get 2 protons for every molecule of FADH. So NADH is actually a little bit more valuable than FADH, but both of them have the same job in that they drop off electrons. The electrons are then used to pump protons. So you end up with a whole bunch of protons up here. There are still protons down here, but there's just fewer of them relative to the top. And so there's some protons floating around in this space. There's a whole bunch of protons floating around in this space. So that's why over here in this picture, if we talk about the charge of the different compartments, the inner membrane space is going to be holding all of those protons. And so the inner membrane space, we say, has a positive charge. And then the matrix, we say, has a negative charge. it doesn't really have any negatively charged molecules floating around. It's just that it's negative relative to the inner membrane space. It doesn't actually have any negatively charged ions. It's just negative relative to the inner membrane space. So we have this difference in charge. And so the product of the ETC, the thing that it makes, is an electrochemical gradient. Meaning that we have a gradient of protons, but because the protons are charged, it also is an electrical gradient. So it's an electrochemical gradient. Or you could also just say it's going to create a proton gradient. Either one of those are probably describing the same thing. You've got this gradient of charge, a gradient of protons, a gradient of different molecules, and all of them are accumulating within the inner membrane space. So once they get there, what do they want to do? They want to go back to the matrix, because the matrix doesn't have very many of them. So just because of normal physical laws of diffusion, they're all up here, they'd rather be down here. So as soon as they get the opportunity to go across this membrane, they're going to want to do that. And that is what is going to happen during chemiosmosis. So during chemiosmosis, so here's the electron transport chain, here's chemiosmosis. During chemiosmosis, you've got another enzyme that is within the plasma membrane, and it's the same membrane. It's also within this inner membrane. And this one is called an ATP synthase. An ATP synthase. And the ATP synthase is a passageway for these protons to go through. The protons can go through the ATP synthase, arrive down here where there are less protons, and it can follow that electrochemical gradient. But if you think about what's going on here, back up to this drawing, you basically have a membrane. On one side of the membrane, you have a positive charge. On the other side of the membrane, you have a negative charge. Well, these two compartments are now being allowed to connect. And if you think about things that you use every day, it's basically the same thing as a battery. You've got one compartment with a positive, one compartment with a negative. When you allow those charges to flow from the positive to the negative, what do we call the flow of charged particles? If you can remember from physics, that is current. That's what current is. And so what we're generating right here is an electrical current. These protons are going through the compartment. You have the flow of charged particles, and so we're generating electrical current. So we've just turned this whole thing into a little electric motor, basically. And we can use that electrical power to drive the formation of more molecules of ATP. And so this ATP synthase is the passageway that is going to connect these two compartments of the matrix and the inner membrane space. It's going to allow the protons to flow from the inner membrane space out into the matrix. And when they do that, that is going to yield electrical energy. The electrical energy we can then use to drive the formation of ATP. And now the whole thing finally is paying off. It's all making sense. Now we're able to use all these molecules we've created along the way, and ultimately the thing that we wanted from that original carbohydrate, that original molecule of glucose, those high energy electrons, is used to pump protons, which are then used to make this electrochemical gradient, which is going to allow the formation of ATP at the ATP synthase. And both of these things are inside of this inner membrane space right here, and that's the reason that the inner membrane has all these folds. If it has all these folds, it's creating more surface area, so then you can pack this inner membrane with as many possible electron transport chains and ATP synthase molecules as possible, so you can crank out the maximum amount of ATP. And so that's the reason why the mitochondria looks that way, and that's also the reason why we're putting all of these protons into this one little space within the mitochondria.